Hi everyone, my name is Shruti Patel and I will be presenting my project today, which is about the evaluation of vector representations of lipid nanoparticles in the cheminformatic predictions of transfection efficiency. And today this is the structure I'll be going through. First, I'll introduce what LNPs are. I'll talk about the background and my question, my hypothesis. I'll go through the procedure of how I made these models, and I'll talk about the data and conclusion. So starting just to introduce myself, as I said, my name is Shruti, and I worked with Lucas as my mentor, and I just want to thank him for mentoring me for these past couple of months throughout this project. I'm presenting from Fremont, California, and I chose this topic because I find the potential of lipid nanoparticles in drug delivery really interesting for advancing healthcare. And this is because there's such a wide variety of applications of LNPs, and there's like many novel areas of research in this topic. And also this will allow me to take a deep dive into the many aspects of LNP research and contribute to the efficient development of LNPs in the future. I also find this topic especially interesting due to their significance in the development of COVID-19 vaccines, which I'll talk about in the following slides. For the background, so what exactly are LNPs? These are tiny lipid-based carriers that are designed for efficient drug delivery, and essentially they can protect and transport different kinds of molecules such as RNA, DNA, peptides, and proteins to specific target cells. And in the COVID-19, the cargo that was transported was mRNAs to create spike proteins in the target muscle cells. LNPs also have anti-degradation properties. These are due to the PEG lipids in their structure, which are hard to break down and allow the LNP to travel further throughout the cell. They're created through microfluidic fusion, in which the lipids are mixed with the drug cargo or the mRNA into the ethanol, and they're self-assembled into a stable nanoparticles for efficient delivery. They're also taken up by the cell via endocytosis, and they're released into the cytoplasm, where they can then enter the nucleus and be translated into proteins. So now I'll go through in detail each of the components of LNPs. So first is the ionizable cationic lipid, which carries a positive charge at a low pH, which allows it to bind negatively to the charged mRNA. Then there's a phospholipid, which is mainly just the structural backbone of the LNP and also contributes to the anti-degradation properties. The PEG lipid is a protective coating, and it also prevents the rapid clearance from the body. It's very stable and increases the circulation time in the bloodstream, and it also controls the particle size and prevents the LNPs from aggregating together while they're in the bloodstream. Cholesterol is mainly used to maintain membrane fluidity and is also used as a stiffening agent. And lastly, the mRNA is what the cargo is. It's in the case of COVID-19, it was the mRNA code to create the SARS-CoV-2 spike protein, and these are protected from all the lipids on the surface of the LNP. So LNPs in COVID-19 vaccine. So I'll just go through really quickly how they were, specifically for COVID, how they were used in the vaccine. So first, the mRNA for the protein was developed in a lab, and then the mRNA was inserted into the LNP, and the LNP is then intravenously injected into the bloodstream of the patient and carried to the target cell. And in the case of COVID-19, the target cells were muscle cells because they're rich in blood supply, so they can create the proteins much quicker and efficiently. Then the mRNA enters the nucleus, where it is then the mRNA can then be turned into the spike protein through translation, and then the spike proteins are expressed on the surface of the LNP, and the immune system is then activated, and the antibodies can then recognize these spike proteins and attack COVID-19 viruses that are found in the body and stop replication. So the data set I used, I found with my mentor, and the data set was for pulmonary gene therapy, and it was pretty recent when I found it. And it was very dense and thorough. It contained over 13,000 LNP activity measurements, and it contained descriptors of the molecular weights and transfection efficiency, which is essentially just how it was delivered, how much was delivered into the target cells. So transfection efficiency was measured through how much mRNA was reaching inside the target cell. So the proteins had this, the fluorescence was measured inside of each protein to measure how much was actually entering into these target cells. And also for when I was creating my models, I shifted all my data by 3.3, which is because to account for the negative values in the delivery efficiencies, and because I re-expressed my data using the logarithm function. So my research question was, what is the best way to represent a lipid nanoparticle formulation in a machine learning model to yield the most accurate prediction of delivery efficiency? Moving on to my hypothesis, out of the components, I hypothesized that if only the cationic and the helper lipids are fully described using the RDKit library, then it will yield the highest unnormalized delivery prediction accuracy. And this is because since the PEG lipid is the same for all of the LNPs and the cholesterol had the same molar ratio in the data set, describing them with RDKit would add just extra noise. So since the cationic and the helper lipids had the most variation in the data, I hypothesized that if those were described the most in the data, or in the model, sorry, then it will yield the highest accuracy. Moving on to my procedure. So before creating these models, I had to prepare the data set for the 3 strategies. So first I imported all the necessary libraries and uploaded them on the data set, as you can see in the bottom left of the screen. And then I used the PubChem website to add the SMILE strings for each of the 3 types of helper lipids that I found. There were only 3 that I found in for all the LNPs in the data set, which were DOPE, DSPC, and DOTAP. And when I was reading through the article for this data set, they only used PEG 2000 for in all of these LNPs, and the cholesterol was also the same. By the way, SMILE strings are it stands for the Simplified Molecular Input Line Entry System, and essentially it translates a chemical structure into a string that's readable by a computer. So if a human was to like look at it, it probably would make no sense, but it's very useful for cheminformatic models like this. Then I created a function that calculates the molecular descriptors, meaning like the molecular ratio or the mole ratios of each of the components, and for the given SMILE string for each of these components. And then I calculated all of these SMILE strings from the cationic lipids, the helper lipids, the PEG lipid, and the cholesterol. Then I normalized all these molar ratios. So the concentrations of the 4 components in total equal 100. So I calculated the normalized molar concentrations into ratios with the given components from the data set. Now moving on to my strategies. So I had 3 strategies in total. The first strategy was every single component. So the 4 components were all fully described, and then they were concatenated into the final formulation vector after they were multiplied by their molar ratios. So this created like a huge data set that was being run through the model, and I predicted that this one would have the most noise, meaning like fluctuations in the data that would hinder the creation of the relationship between these variables. The second strategy was only fully describing the cationic lipids through RDKit, and this one I predicted would have a lower accuracy because only predicting the cationic lipids would leave behind the other structures in the other components in the structure of the LNP. And then lastly, strategy 3, which was my hypothesis, the cationic lipids and the helper lipids were fully described. And as I mentioned earlier, this was because they had the most variations in the data set as I was studying them. So now moving on to the data and results. After I fully trained all these models. These are the metrics that I use. I studied the R squared, which is essentially just how related or how correlated the two variables are, and then the mean error, which is the lower it is, the better it is. That means that it's more accurate. The model is more accurate. So for strategy 1, the mean absolute error I got was 1.6, around 1.611, and this is not too low compared, or not too high compared to the other strategies. So I was pretty surprised that it wasn't as bad as strategy 2, which was 1.7, which was a lot higher than the first one. And in this strategy, just as a reminder, it was only the cationic lipids were fully described and multiplied by their mole ratios, whereas all the other components were fully added added to the formulation vector. Then for strategy 3, the mean absolute error was 1.5, which was the lowest out of all of them. And it also had the highest R squared. And this supported my hypothesis. And just a summary, just to put them all side by side so you can see the difference. For strategy 1, it was a 1.6, strategy 2 was slightly higher, and strategy 3 was the lowest of all of them and also had the highest R squared. So now moving on to my conclusion. My hypothesis was supported as strategy 3, which was the cationic and the helper lipids were fully described, yielded the lowest error. So I concluded that it was more accurate than the other formulation vector construction strategies. Strategy 1 likely had too much noise, which is random or unpredictable fluctuations in the data that disrupt the ability to identify target relationships. And strategy 2 was likely not descriptive enough. And so what does this indicate? It indicates that fully describing the molecular structures may be likely is to be more effective to capturing the relevant features of LNP formulations, so that if the model used detailed and chemical descriptors for these two lipids, it can capture their role in delivery compared to models that fully describe the cholesterol and PEG, or those that use only basic formulation data. So essentially, a simpler or more computationally efficient strategy could be used without sacrificing efficiency in the model prediction. And essentially, this is practical for developing new LNPs for different kinds of diseases or viruses, which is it'll lead to LNP development that is much more efficient and quicker, which is really essential for pandemics and any types of disease such as COVID-19. And further investigation could be, I could study other variables instead of just this component. So it could be particle size or other biochemical properties. Or I could, instead of studying descriptors, if I could find a data set explaining other representations, that would be useful as well. Thank you. Thank you everyone for listening. That's all I have. Are there any questions?
Thank you so much, Shruti. I just want to say I'm so impressed with like all of the importing of data sets and calculating and training models that you did. That's a ton of work. So congratulations, just first and foremost.
Yeah, thank you.
Absolutely. I'm curious, what would be kind of your advice to a student who wants to do kind of a similar project or is interested even just like in the broader field of drug delivery?
Yeah, if you're interested in studying drug delivery, I think it's like a really interesting topic, especially nowadays, since there's so many new technologies that can be used in drug delivery, making it more efficient. I think it takes like a lot of research to just find different kinds of like data sets if you want to study those. And obviously I couldn't do this without my mentor, so he also helped me with teaching. Like this was something new that I haven't worked on, like using machine learning for this kind of topic was like pretty new. So I would recommend like watching like a lot of videos online and reading articles about how to use these kinds of models to enhance drug delivery systems.
Very cool. Thank you so much.